Hi, so my name is Hannah Powella, and I studied the effects of RECQL overexpression on herpes simplex virus type 1 infection. So, herpes simplex virus, also known as HSV1, affects 50 to 80% of the general population, and typically symptoms are presented as cold sores or fever blisters, but can also progress to more severe effects such as keratitis, leading to blindness, or also encephalitis. So we know that multiple viral and cellular proteins act on the viral genome during HSV1 replication, and we found that human RECQL-like helicase, which is RECQL, associates with the HSV1 genome during viral DNA replication. We know that RECQL is a DNA helicase enzyme and it repairs DNA damage and maintains a stable genome, and that the function of RECQL during viral replication has yet to been defined. Prior research in our lab found that RECQL knockdown resulted in a slight increase in viral yield, which led us to our hypothesis that RECQL is inhibitory during HSV1 replication, and overexpression of RECQL will result in less efficient replication of HSV1. So, first to test this, we cloned RECQL into the PLEX-C tag vector, and we transfected into HEC293 cells. Proteins were then collected to determine expression of RECQL by Western blot, or our cells were infected, which HSV1 caused, and 24 hours later, the virus was collected. HSV1 yield was then measured by a plaque assay. So first, I was able to successfully clone RECQL into the PLEX-C tag. Here in Figure 2, we're able to see a PCR confirming the presence of RECQL in the vector, and also see a diagram of the vector on the right. After we transfected RECQL into our HEC293 cells, we wanted to determine if we successfully overexpressed RECQL. And here in this Western blot, we were able to see that it was successful. And finally, we infected our cells with HSV1 and collected the virus. And we wanted to see the HSV1 viral yield with overexpression of RECQL. So here, our hypothesis thought that RECQL will result in less efficient replication of HSV1, but in the graph on the right, it is clear that there's no discernible change in the viral yield with overexpression of RECQL. So in conclusion, RECQL was first successfully cloned into the vector, and then it was also overexpressed in cells. However, the plaque assay indicated that even with an overexpression of RECQL, the replication of HSV1 was not altered. Therefore, we accepted our null hypothesis. In the future, I would like to repeat this experiment with different concentrations of RECQL to determine if the effects on HSV replication is dose dependent. I would like to thank my principal investigator and mentor, Dr. Jill Dembowski, and the entire Dembowski lab, along with Duquesne University School of Science and Engineering and the Department of Biological Sciences.